If you're a little bit rusty on the female anatomy and talking about the follicles and secondary oocytes and things like that, you might take a minute and just review that a little bit before we get started, because we're going to take some of those structures and those concepts that we learned about in chapter 22 and apply them to the pregnancy and embryonic development today. Okay, so as we learned in chapter 22, spermatogenesis is the production of sperm, and oogenesis is the production of the oocyte or the egg. And those are, the sperm and the oocyte are the sex cells or the gonad, the gametes that are produced for reproduction. And remember, a secondary oocyte is going to be surrounded by follicular cells. And when it's ovulated, it's called the Graafian or mature antral follicle. So as you probably already know, fertilization is a process where the sperm and the oocyte unite together and form a zygote. Okay, so remember, the sperm and the oocyte both have 23 chromosomes. They're what we call haploid. So when you unite those together, you get a diploid or a 46 chromosome zygote. Okay, and so at that moment when that happens, that zygote is a unique person. It has its unique genetic information that's different from mom and dad, but yet comes from mom and dad. Okay, but they're a genetically unique individual when they form this single cell, cell zygote. Okay, and then pregnancy begins at that point. Now, in this next bullet point, it shows us that after 38 weeks of cell division, the baby is born. Okay, now if you have gone to the OB, or maybe you have a friend or know someone that's been pregnant and they talk about 40 weeks of gestation. This 38 weeks here is from basically fertilization to birth. When you go to the OB doctor and you're talking about the 40 weeks of pregnancy, they add 2 weeks on there because ovulation usually occurs about 2 weeks or 14 days after the beginning of the last period of the menstrual cycle. And so it's really easy to pinpoint when the period started. And so that's why the doctors will start 2 weeks back and count from there, because it's a really good place to pinpoint, because you don't always know exactly when you have ovulated. So that's the difference there. Okay, so this chapter is talking about growth and development, and some of you that are pre-nursing or pre-healthcare have taken human growth and development, and you've probably studied way more about this. But growth is an increase in size. Okay, so we go from a single cell zygote to a 7-pound baby. Okay, so that's going to increase in size, but it's also going to develop. And basically, development, which includes growth, but it's a process by which an individual cells and individual tissues and organs change from one phase to another. Okay, it's going to go from stem cells, which are undifferentiated or basically kind of like generic cells, to very highly specialized cells like neurons and muscle cells and epithelial cells and things like that. So pregnancy is going to involve 2 periods, or I'm sorry, not there yet. Life involves 2 periods: the prenatal period, which is from fertilization to birth, and then the postnatal period that we're all in is basically your life after birth until death. Okay, so we'll talk about next time about aging and actually the process of dying, because it's all part of life. And we're going to start here though today with the process of fertilization, and it's also called conception. And that's going to take place, as we learned in the last chapter, in the uterine tubule, okay, and perhaps even in the infundibulum. Remember, we have the fimbria and we have the funnel of the infundibulum, and then we have the uterine tube that connects down into the uterus. And then the ovary will be like right here. The egg is released, the sperm will come up, and they will unite in the fallopian tube, or the uterine tubule. Before we move on there's some things that need to take place before fertilization can happen. So must take place before fertilization. One thing that has to take place before fertilization can happen, obviously, is ovulation. And the egg has to enter the tube. Okay, now sometimes you can have fertilization take place outside of that tube, and that results in an ectopic pregnancy, and it's life-threatening to the mother. Number two, so once you've ovulated and that egg enters the tube, okay, this is another no-brainer. The sperm must reach the egg. And in the last chapter, we learned about some of the secretions that make up semen that help the sperm survive and be nourished on their journey. Okay, so there's those secretions. There's also prostaglandins. That are released in semen, which are paracrine secretions, if you remember that term from chapter 13. Those prostaglandins in semen stimulate, or basically signal the sperm to swim. Okay, they're like, it's like a little cheerleader, cheering them on, trying to get them to go. And then estrogens from the female help to thin the female secretions, and it's going to help with sperm motility. Okay, it's going to help them basically swim through those secretions and swim from point A to point B a lot more easily. Later on, as you get farther away from ovulation, the woman's body secretes progesterone, which will thicken the secretions and inhibit sperm motility. So we talked about the ovule must be ovulated, okay? And this oocyte is going to last only about 12 to 24 hours after ovulation. So if it is not fertilized within that very short time period, it's no longer viable and cannot be fertilized. Now, in this diagram, here would be the maximum point at which the sperm could be deposited in the body. And so they have to make a journey all the way up through the uterus, through the uterine tube to the site of where the oocyte is going to be. Half of the sperm are going to go to the wrong direction. Remember, it's a very acidic environment. They have to make it through the cervix. And so most of the sperm are not going to make it and are going to die en route. So only a couple of hundred out of the millions, 200 to 600 million, are actually going to make it. And you can see in this diagram here, the little blue areas are sperm trying to penetrate the corona radiata of the follicle there. So it takes them about a half an hour to reach the egg once they have been deposited. And it's best if they can fertilize that egg right away. Okay, usually sperm can live up to 6 days, but remember, it's a pretty hostile environment, and it it's best done if fertilization takes place within the first 24 hours. And the sperm in the video that I'm going to have you guys watch, you can see like they'll kind of die down and lose some energy and become a little bit dormant. And then there can be situations where if ovulation takes place and the sperm are present, there are hormones that can reactivate them and reinvigorate them and have them go and try to finish off the rest of the journey. So they can live up to 6 days in the female reproductive tract. Okay, so the video that I'm going to have you watch that's required, and you will watch for points, is going to show you how the sperm actually penetrate the different layers of the follicle and then penetrate the oocyte itself and unite with the nucleus of the oocyte. And if you want to look at this diagram here while I'm describing this, that might help. But remember, there are two layers around the oocyte itself. And so let me just show you. So here's the actual secondary oocyte, this portion here. Then we have the white area, which is the zone, or the blue area, which is the zona pellucida. And then we have the corona radiata, that are these extra cells out here on the edge. And so the first thing they have to do is basically burrow through the corona radiata. Now, remember the structure of the sperm. You have the tail, which moves back and forth and helps them swim. This mid piece is where all the mitochondria are, and ATP is produced to power the tail. And then the head has an acrosomal cap. Do you remember that? It has enzymes that help burrow through the zona pellucida. And then the blue part here is going to be the nucleus that carries the genetic material that's going to unite with that oocyte. So several hundred of the sperm are going to start to use their enzymes in their acrosome cap to start dissolving the follicular cells around the outside. And then they're going to start to basically dissolve the layers through the zona pellucida as well. They have enzymes that can do that. There's hundreds of sperm that can actually start working on those outer layers. Okay, but when you get to actual fusion of the sperm and the egg, only one sperm can actually penetrate the oocyte's membrane and fertilize. Okay, if there are if two sperm accidentally penetrate the oocyte's membrane, the oocyte is inviable. Basically, it means it's not going to divide and become an embryo. So only one can actually penetrate and fertilize. In the video, I'll show you what happens to the zona pellucida and other structures as that initial sperm penetrates. Okay, so once the sperm has entered into the membrane or through the membrane of the oocyte, the sperm nucleus basically triggers the oocyte to finish meiosis. Okay, so let me jump ahead a couple of slides here. So meiosis continues once the sperm comes in. Okay, remember we said in the last chapter that for the secondary oocyte to finish meiosis 2, that sperm has to fertilize that egg. If that egg is not fertilized, then it will not complete the meiosis cycle. And so it's kind of, the oocyte is basically in pause here. It has not finished this meiotic division. And so once that sperm penetrates here, the oocyte will go ahead and finish meiosis, produce that first polar body, which remember it disintegrates. And then the oocyte nucleus is ready for fusion, where the sperm nucleus and the nucleus of this cell fuse together and form that zygote. One other thing that you might notice as well is that the sperm does not completely enter the cell. Only the headpiece enters the oocyte. The midpiece and the tail remain on the outside. Okay, so at the point right before they unite, the nuclei of the sperm and the nuclei of the oocyte each are called pronuclei. And then once they fuse, when they unite, that becomes the zygote and has 46 chromosomes, which is how many chromosomes a human cell has, except for the sperm and the egg. Okay, so that's the process of fertilization. It might be a good idea right now to pause this video and watch the fertilization video on D2L and kind of get a visual of what is happening. Okay, so once fertilization takes place, pregnancy is the next step, and pregnancy is defined as the presence of developing offspring in the uterus, and it is divided up by the doctors into 3 trimesters, and each trimester is about 3 months long, or about 12 weeks long, and it is also divided up into 2 stages. The embryonic stage, which is the conception or fertilization to 8 weeks. And then the fetal stage is basically 9 weeks to birth. So make sure you know the designations between embryonic and fetal stage. So if I give you some characteristics, you can be able to tell me, is this an embryonic stage, or is this happening in fetal stage? And I'll kind of give you some of the main milestones that happen during those time periods. Okay, so embryonic stage, some main characteristics. Okay, the placenta is going to form. The main internal organs are going to develop during this time period. And also you're going to start to see some external body structures like arms and legs and the head and the nose and things like that. So about 30 hours after fertilization, the zygote, which is basically egg and sperm that has formed together, they've united their nuclei into one. The zygote, after 30 hours, is going to undergo mitosis. Remember, meiosis makes the egg and the sperm. Mitosis, cell division, allows for growth and development. So on this next slide, we're seeing here is the secondary oocyte. And there are, these little guys are sperm. And these are follicular cells. Here's the secondary oocyte there. And then this is going to be the zygote's first division, first mitotic division. And this is what we call a morula. And it's a ball of about 16 cells. Okay, so during this time of cell division, we have what is called cleavage. And that's where these cells divide and the cells themselves become smaller and smaller, but more numerous. Okay, so we're going to go from 1 cell, it's going to divide into 2, and then to 4, and then those are all going to divide into 8 and 16 and so forth. But during cleavage, the size of the ball of cells, or the blastomere, doesn't increase. So each of these small cells here, when they divide, they kind of divide in half and they become progressively smaller. Like the whole mass of cells does not get bigger during the cleavage phase. Okay, so here's some definitions that you'll want to know. So the zygote's the fertilized egg. The morula is a ball of cells, and it's about 16 cells. And then we're going to talk a little bit about a blastocyst here. But before we do that, the morula, so the zygote is fertilized in the uterine tube. And last time we talked about how the uterine tube has cilia and has weak peristalsis to kind of push the zygote down toward the uterus where it implants. If it implants in the uterine tube or anywhere else in the body besides the uterus, that's when you get that ectopic pregnancy that I mentioned before. And so the morula is going to take about 3 days to travel down the tube using the tube's cilia and peristalsis. Okay, now as the morula continues to divide, okay, it's about 16 cells at this point, but those cells are going to continue to divide and go through mitosis. And so the next stage of embryological development is going to be the blastocyst. And this morula is a solid ball of cells, but the blastocyst is a hollow ball of cells. And this hollow ball of cells at about 6 to 7 days after fertilization are going to then implant in the uterus. Okay, so you have fertilization, about a week later, you'll have implantation into the uterus. Okay, so the blastocyst is going to be made up of 2 different layers. It's a hollow ball of cells, and I'll show you a picture of it here in just a second, that's made up of the inner mass, okay, and this inner mass, I'm going to come up here, is also called an embryoblast. And the embryoblast gives rise to what's called the embryo proper, or basically this is the part of the embryo that becomes the fetus. Okay, so the whole embryo doesn't become the fetus, okay? Only this inner layer continues to develop into the baby. The trophoblast is going to be the outer portion of this embryoblast structure, and it's going to be used to form assisting structures like the placenta, okay? The placenta, as you know, after birth is discarded, okay? It's not a part of the baby, okay? So it is necessary, though, to connect baby or embryo or fetus, depending on the stage, to mom to get nourishment, okay? And that's where it's going to come from. It also is going to produce the hormone HCG, human chorionic gonadotropin, and that's the hormone that's detected in like the home pregnancy tests. And HCG is going to be important to maintain that corpus luteum. Remember, after the oocyte is ovulated, the remnant of the follicles remain there, and they're going to secrete hormones to help maintain that pregnancy and not continue on with the menstrual cycle. So the trophoblast is really important because it's going to help form the placenta and other structures that are going to help babies survive. And then it's also going to produce the hormones that are needed to maintain that pregnancy. So the blastocyst is made up of the trophoblast and the embryoblast, and the embryoblast is the part that actually becomes the baby. Okay, so here is just a diagram of all of this. Okay, so remember the pronuclei is the egg and the sperm nucleus before they unite. Once they unite together to form the zygote. So 23 plus 23 gives us our 46 chromosome zygote. about 30 hours, the zygote's going to divide. Okay, this is the first 2-cell stage, 4-cell stage, morula. Just remember about 16 cells, and it's a solid ball. And you can see the, excuse me, the cleavage taking place where the cells are dividing, but they're not growing in size. Okay. And these are all called stem cells. And so maybe you've heard of stem cell research before. Stem cells are what we call undifferentiated cells. And they're basically kind of like generic cells that they just divide and divide and divide. And then later on, they'll become specialized. Okay, so these are unspecialized cells at this point. Okay, so we have ovulation, fertilization. Okay, 30 hours later, we start to get this division. And then 6 to 7 days later, the blastocyst will implant. Now, I said the blastocyst was a hollow ball of cells. This guy here, okay? So here's the stem cells, which are going to be the embryoblast. And these outer cells are going to be the trophoblast. Trophoblast, you can see those brown cells, they're digging into the endometrium of the uterus. Okay, that inner lining of the uterus there. They're going to help form the placenta. And the blue cells, the embryoblast, is going to go on and form the embryo or the fetus or the baby eventually. Okay, so here is that embryoblast that is starting to implant. So it'll first attach, here's the endometrium of the uterus. Okay, here's the endometrium layer here. And that's the part, remember, that shed during menstruation if pregnancy doesn't occur. We're going to attach here. This is the blastocyst. The trophoblast cells are going to attach. And then the trophoblast cells are going to actually kind of invade the uterine wall. And it embeds those cells into the endometrium. And we get then what's called implantation. And implantation is basically when the blastocyst kind of like is burrowed down into the uterus. So you can see here, the endometrium is actually surrounding the embryo, and it's completely covered by that endometrial lining. But soon after this happens, the trophoblast enzymes start to break down the endometrium or the surrounding uterus area there for nutrients. And this happens until the placenta is fully formed. Okay, so implantation begins at the end of the first week, day about 6 or 7 after fertilization, and then it's going to conclude and be complete around sometime in the 2nd week. Okay, so here is a drawing of this. Here's the endometrium, all this kind of light tan pinkish color here. All of that is the endometrium of the uterus. And then all these little squiggly lines here and everything in there is the forming embryo. Okay, so as the cell, the embryo cells are dividing, they're forming some different layers. And this layer here is the trophoblast layer, but there's another layer that's starting to form just inside of the trophoblast. And if you, which is this here, and if you join those together, that's called the chorion. And the chorion, they call it, is the outermost extra embryonic membrane, or outside of the embryo membrane. Okay, so this is basically the baby developing right here. And this is going to fuse together and form a membrane outside of that, and that's called the chorion. Also, the trophoblasts are going to start to send out projections into the endometrium of the uterus and digest parts of the endometrium, like we said, for nutrients. And these little fingers that are coming out here are called chorionic villi, and they're going to be pretty well developed by the end of the 4th week after fertilization. Now, as the chorionic villi from the trophoblast to begin to digest parts of the endometrium, they form these little cavities called lacunae. And I'm going to show you a picture of those here. I think here. So here's mom's blood, the arteries and the veins in the uterus. Okay, here's baby beginning to develop the placenta with its blood vessels here. This area all along here in between, that's kind of a purplish color, those are lacunae, and those are areas where nutrients diffuse out of mom's blood, like oxygen and glucose and amino acids and so forth, and are picked up by baby's blood. Okay, now there's things coming from baby, like carbon dioxide and other wastes that are going to get picked up by mom's blood. Okay, and so this is the beginning of the formation of the placenta. Mom's blood and baby's blood never mix. Okay, but these spaces in here that have been dissolved by the chorionic villi cells or the trophoblast cells are called lacunae. Okay, they're going to fill with blood and supply the baby with some nutrients. Okay, let's see. Embryonic blood vessels are going to transport body fluid, nutrients and so forth from the embryo into the developing placenta and exchange gases and so forth with mom. Let's see. Then we have another layer called the amnion, and it's going to be also another extra embryonic membrane, or outside of the embryo membrane. And it's going to form while the placenta is forming. The chorion is the layer that's going to form the placenta. And then again, we're going to have the embryonic disc, which is basically the fetus. Okay, it's the embryoblast, but now it has developed into an embryonic disc. Okay, it's developed a little bit more from what we've seen it before. And you've maybe heard of embryonic fluid before, or maybe a pregnant woman says, oh, my water broke, or my bag of waters broke. That's the amniotic fluid, and that's basically fluid in between the embryonic disc and the amnion. Okay, and its job is to cushion the baby, keep the temperature where it should be, and keep the outer structures of the embryo from drying off, or the fetus from drying out. And then we have the umbilical cord. Okay, the placenta and the umbilical cord are two different structures. The umbilical cord is basically a structure that encloses the blood vessels. Okay, the umbilical cord basically connects the placenta, which is connected to the uterus of the mom, to the umbilicus. Okay, the umbilicus is basically what your belly button formed from. Okay, so I'm going to come back to this slide here. This shows the 3 germ layers. And we're going to talk about those a little bit more here in a minute. Where was that? This embryonic disc is formed. is the embryonic disc here. And that disc has 3 layers, and we'll talk about those 3 germ layers here in just a few minutes. I'll come back to that one. Okay, so amnion, this outer membrane here, and the chorion is going to come together and help form the placenta. The connecting stalk is going to form the umbilical cord as that baby is developing. Okay, so the placenta, this is really important. I want you guys to star this whole slide, okay, and understand the makeup of the placenta and the umbilical cord, okay? After we watch next time's video, we're gonna have a case study on fetal blood flow, okay? So it's gonna be really important to understand the structure of the placenta and also the umbilical cord. Okay, so the umbilical cord includes part of the embryo, okay, the embryonic portion or the baby's portion, and also a maternal portion, okay? So here's the myometrium, which is the muscle layer of the uterus, okay? And the endometrium, okay, is going to be that inner layer of the uterus. And the placenta, here's the lacunae, okay, basically burrows down into that and attaches into the endometrial wall, okay? So maternal blood's gonna come up here, the mom and baby blood do not mix. There's diffusion of nutrients, just like there's not blood that fills the lungs, right? The blood stays in the capillaries and the gases exchange across the cell walls. Same thing here, okay? So maternal blood will fill these cavities, but the gases will exchange across, okay? So there's no blood mixing. But here's another important thing that I want you to remember. The umbilical cord contains 3 blood vessels, and you can see those here. There's gonna be 2 umbilical arteries and 1 umbilical vein that's gonna bring blood from the embryo or the fetus to the placenta and back, okay? Now, take note here. Typically, when we think of arteries, we think of oxygen-rich blood, right? Okay. Remember though, arteries take blood away from the heart. Veins, V-E-I-N, takes blood into the heart, okay? With the fetus or the embryo, we're talking about the embryo's heart, not mom's, when we're talking about this umbilical cord. So here, we have 2 umbilical arteries, 1 umbilical vein. Look at here, the vein is red, okay? That is because that umbilical vein is going into the baby's heart and it's taking oxygen-rich blood from mom, from the placenta into baby's heart, okay? So that vein is going into the baby's heart. The umbilical arteries are taking deoxygenated blood out of baby back to the placenta where it can pick up oxygen. Okay, so it's a little bit backwards of what we normally think of veins and arteries on. Okay, so if you need to pause here in a minute and contemplate that and think through it, go ahead and do so, because that's one of the things that I want you to remember and get stuck in your head. Okay, this is on page 879 in your textbook, if you want to get a little better view of that besides what's in your handout. Okay, so during this whole time and we're in embryonic development, we're still in the first 8 weeks past fertilization, a lot of the structures are going to develop, and then within a few days or weeks, they're going to change again. Okay, so we have a time when the amniotic cavity gets so large that it's going to come in contact with the chorion. And then those 2 membranes, those 2 layers are going to fuse together and form the amniochorionic membrane. And I don't see a picture. Okay, so this is going to fill, and the amnio and chorionic membrane, those are going to fuse together. Okay, then there's a yolk sac. It's going to form blood cells. The atlantis is going to form blood cells as well and help form blood vessels in the umbilical cord. The placenta, we've talked about that. It's shaped like a disc, and it's going to be where the chorion contacts the uterine wall or the endometrium. And then the placental membrane is basically the site of gas exchange. Okay, it's a membrane, it's a place in between maternal and fetal blood so that they do not mix. Okay, now the placenta, sorry, is about 1/2 inch long. I'm sorry, 1/2 inch thick. And about 10 inches long. Okay, and it just attaches and kind of forms, sorry, get in the view here, forms right in with the uterus. See if we can find a good picture. Maybe not. Sorry, I should have looked for this. Oh, here we go. So this structure here is the placenta. It's pretty thick, about 1/2 inch thick and 10 inches long. And then here's the umbilical cord with the 1 umbilical red vein and the 2 umbilical arteries. Okay. What else did we have here? Oh, the placenta is also going to be not only a site of gas exchange, but it's going to be really important for the production of progesterone, a hormone. That progesterone is going to get converted by the body to estrogen. And those are going to cause changes in the mom. Okay, they may we'll talk about maternal hormones later on, but things to help with mammary gland development to get ready for nursing, relaxation of the pelvis and other ligaments and things like that, getting ready for birth. And progesterone is very important for maintaining a pregnancy as well. Okay, I think we caught everything on that slide there. So here's just a diagram of the placental membrane. Okay, so here is the placenta. Okay, the uterus will be kind of attached down in here, and then baby would be out here. Okay, so we have the 2 umbilical arteries, the 1 umbilical vein. If we cross-section this off here and take a look at it, here are the capillaries to baby's veins and arteries there. The lacuna, just filled with mom's blood here. And then, so do you see there's basically a separation here? Okay, that's the placental membrane. And if there's oxygen here, we'll diffuse across there and get into the umbilical vein. If there's carbon dioxide, will come out and go back in here so mom can take that waste away. So you have the diffusion of gases and nutrients across that membrane. Okay, now after the embryo implants, okay, we get a second stage of development called gastrulation. And that's where you start to get the embryonic disc forming and forming those multiple layers that I was talking about. So you still have like the trophoblast extra embryonic cells, and then you have the embryonic disc, which becomes the baby. Okay, so at the end of the second week after fertilization, you have you have two layers, the endo and the ectoderm. The outer layer is the ectoderm, the inner layer is the endoderm. And then later on, you get a third layer develop in the middle called the mesoderm. Okay, and these three, what we call primary germ layers, is going to form the major internal organs. And this is called organogenesis. It's basically the layers from which things like the kidneys, the skin, the skeletal system, and all that develops from. It's called organogenesis. So an embryo that's past gastrulation has those three primary germ layers. And let's back up to, should move this, sorry, back up to this slide here. Okay, so here's our three germ layers. The endoderm, this yellow layer, the inner layer, is going to form things like the respiratory and digestive systems. The mesoderm is going to form the heart, the muscles, urinary system, and the skeletal system. And then the ectoderm is going to make the skin, the nervous system, and so forth. Okay, so these layers of the embryo, they're stem cells right now, they're very generic cells, but as development continues, they will divide and basically become specialized cells. So if they're in the mesoderm, some of them are going to become the kidneys. If they're in the endoderm, some of them are going to become the liver, and so forth. Okay, so let's see. Here's a really good chart for you guys to look at, because I do want you to know the different stages of embryo development. Okay, so know what a zygote is, know what cleavage is, know about during which days or weeks these occur during the development stages. Okay. Oh, so here this just continues on with gastrulation and organogenesis. Ecto and endoderm cells are epithelial cells. Mesoderm are connective tissue. During the fourth week, past fertilization, you get a neural tube, which becomes the spinal cord. The head and jaw appear, the heart starts to beat, and the limb buds, or little baby arms and legs, start to develop. Okay, so here's some embryos at 3, 3.5, and 4 weeks. And these structures here are part of the extra embryonic structures. So this is the embryo. This is the embryo that's going to become the fetus portions. And this is the developing placenta. Okay, so gastrulation brings us through the fourth week, week 5 through 7. Oh, and you can see the little arm buds here and the umbilical cord attaching here. Here's a little guy. Isn't he cute? He or she is, let's see, about 7 weeks. Is this little person right there? Okay. One thing you might notice too is that there is a tail. It's called a post-anal tail, and it regresses by the 8th week. Okay, so the limb buds are developing. Here's the heart. Okay. Let's see, the head grows really fast in comparison to some of the other structures as well. But by the end of the 7th week, most of the internal organs have been established. They're obviously not fully developed and ready to go and going to work properly 100% at this point, but they're all there, okay, and ready to continue on with developing. Okay. There's also an embryological development video that I'd like for you guys to watch as well. So make sure you check that one out and watch it for points. I can't write today. Okay, one thing that I wanted to talk about right at the end here, and this is an 8-week embryo, kind of that borderline between embryo and fetal stage, because remember, oh, excuse me, oh, the 9th week is going to be the fetal stage. But during the first 8 weeks is very, very important for that embryo, because there's a lot of cell division going on, and if something gets disrupted with cell division, that can cause some problems. Okay, so you see the reproductive system, when those structures are developing, by the 4th week, most of these things are all here. Okay, the heart's very well developed. The central nervous system is going to continue to really develop and become more and more specialized and developed. But there are what we call teratogens that can, or basically chemicals, any kind of factor that can cause malformations or cause problems during embryological development. And the embryo is very, very susceptible to these drugs or these chemicals during fetal development because there's so much growth and development occurring. So here's some examples of teratogens. There could be drugs, x-rays, viruses. Okay, so when you go to the dentist, if you're a female of reproductive ages, or you just go and get an x-ray in general, they'll ask you first if there's a chance that you could be pregnant, because if you are, it's not good for the embryo or the fetus to be subjected to those x-rays. Okay, so Accutane, maybe you've heard of that before. It's used to treat acne. Okay, if a person is finds out they're or maybe doesn't know they're pregnant and they're taking Accutane and they get pregnant, that can cause some significant birth defects. Let's see, diethylstilbestrol. This is a synthetic estrogen. And that was actually given from the 1940s to the 1970s to women to prevent miscarriage. And it actually caused more damage. Thalidomide is another one. It was given in the 60s and 70s to women that, and actually we can switch to this one, was helped with nausea. It was a kind of a tranquilizer if mom was very anxious or nervous during pregnancy, and it would cause some serious limb malformations, like the limbs wouldn't be developed properly at all. Rubella, like the MMR vaccine that we get, rubella can, the reason we really vaccinate for that is to prevent birth defects from that. Alcohol, cigarettes can cause a lot of problems, occupational hazards, and so forth. Okay, so I'm going to stop with our lecture from there and continue on with this chapter in the next lecture. So check out the daily checklist for your assignments. You have a couple videos, and I believe there will be a Connect homework. I haven't completely decided on that. So when in doubt, always check the daily checklist for what you need to work on, and also make sure that you are Double checking your grades from Connect into D2L. I have been very overwhelmed by grading and homeschooling kids and different things like that. And so I have made a couple of mistakes. Some of you have caught me on that, and I appreciate that so much. So please bear with me. I'm not trying to fail anyone. So please double check because I have made some ridiculously dumb mistakes with grades. So please help me with that and make sure that I'm not giving you a 10 instead of a 90 or something crazy like that. Okay, so I don't do that on purpose, but things are crazy here. So again, if you need anything, let me know. I think you should be able to go in and look at your tests and see how you're doing on those, how you, which ones you missed, what the correct answers were, and so forth. And please, 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 we're getting close to the final. Start today looking over your notes all the way back from the introductory lesson through the blood chapters, the heart, cardiovascular, all of that stuff, because there's a ton of information. It is cumulative. And so please, please, please make sure that you're starting to review your notes and get ready for the final today so you can be successful. And if you need any help with that, just let me know. All right. So hopefully you guys are doing well, and we will talk to you again soon.